All right, in this next section, we're going to talk about the origins of photosynthesis and ATP. First, photosynthesis. Ancestors of Cyanobacteria, prokaryotes, were the first cells that carried out photosynthesis pathways using the sun's energy. Over time, these pathways became more complex and efficient, leading to today's eukaryotic photosynthesis. Today, the ocean is filled with different types of these blue-green bacteria. They grow in string-shaped colonies, which you can see in the images. What is the impact of the Cyanobacteria ancestor? Prior to 2.45 billion years ago, there was an insignificant amount of oxygen in the atmosphere. The rise of Cyanobacteria's ancestors carrying out photosynthesis supplied abundant oxygen, which enabled the evolution of eukaryotes and then larger forms of life in the Great Oxidation Event. Some of the Cyanobacteria ancestors entered into a symbiotic relationship with plant cells and exist today as chloroplasts inside the cells. Evolution and metabolism. Early prokaryotes used pigment and its properties to trap light energy and utilize that light energy to do work. They became more abundant because they had a newfound infinite energy source. As they became more abundant, new variations began to emerge with more efficient ways to utilize the energy source. Prokaryotic photosystems are the first organic systems to turn energy from sunlight into chemical energy. They are a conglomeration of proteins and pigments that have the ability to use enzymatic activity and resonance transfer to absorb sunlight and use its energy to produce a molecule of ATP. A ribbon diagram of a photosystem is shown. Proteins are chains of amino acids that fold up on each other to produce useful functions. ATP, or adenosine triphosphate, which you see in the image here. Here's the adenosine and 3 phosphates, is the currency of energy in living systems. It stores energy that's gained from exergonic reactions, and then it uses that energy to power endergonic reactions at a later time. ATP provides the energy for the processes of life. Cells can store the energy from catabolic pathways in a molecule called ATP, adenosine triphosphate. Catabolic pathways are those pathways that take big molecules and break them down into their smaller building blocks. Okay, so when those big molecules break down into smaller building blocks via catabolic pathways, the energy stored from that can be stored as ATP. Then the ATP can be broken down later to fuel anabolic reactions, which is the reverse of a catabolic pathway where building blocks are synthesized into larger molecules. ATP is a nucleotide consisting of a nitrogenous base, adenine, a ribose sugar, and 3 phosphate groups. So we have adenine, the ribose sugar, and 3 phosphates. It's similar to DNA and RNA nucleotides, but ATP has 3 phosphate groups and DNA and RNA only have 1. ATP adenosine triphosphate. So this is a common name, right? Adenosine is the combination of the adenine and the ribose sugar. Adenosine with 3 phosphates. Triphosphate includes 3 phosphate groups, which you can see circled. Each phosphate group has an ionic charge of -3e. In this model of ATP, each phosphate molecule is circled in, or phosphate ion is circled in blue. E is just the charge on an electron, so each phosphate group has an overall -3 electron or elementary charge, which is 1.6 times 10 to the minus 19 coulombs. The phosphate groups repel each other since they each have a negative charge. Therefore, it requires work to add the second phosphate group to go from AMP monophosphate to ADP diphosphate. To add the third group to go from ADP to ATP triphosphate requires even more work since it is repelled by both of the other phosphate groups. This is like the work in compressing a spring. The energy from the work needed to bring each phosphate group to the molecule is stored in that phosphate bond. When the bond is broken to go from ATP to ADP, significant energy is released. Going from ADP to AMP releases less energy since there is less total charge in ADP than ATP. The bonds between the phosphate groups of ATP's tail can be broken by hydrolysis. Energy is released from ATP when the terminal phosphate bond is broken. The released energy is equal to the work that was done to form the bond. That work overcame the electrostatic repulsion between the last phosphate group and the initial ADP molecule. The result is a chemical change to a state of lower free energy. In living systems, the energy from the exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction. Overall, the coupled reactions are exergonic. The early prokaryotes used the energy stored in ATP to run their life functions by converting it to ADP. If you're interested, you can click here for more resources about ATP. All right, let's take a closer look at how ATP performs work. ATP can drive endergonic reactions, those reactions that require an input of energy, by something known as phosphorylation, or transferring a phosphate group to some other molecule, such as a reactant. So if this is my reactant, look what happens here. On the other side of this, it now has a phosphate group attached to it. This recipient molecule is now what we would call phosphorylated. 3 types of cellular work are powered by the hydrolysis of ATP. So what's happening to the ATP here? A water molecule breaks the bond between the third and second phosphate, converting ATP into ADP. So now this is adenosine diphosphate with only 2 phosphate molecules. There's 2 here, and the third is now on this recipient molecule. The 3 types of cellular work powered by this hydrolysis are transport, mechanical, and chemical. So again, the hydrolysis of ATP is an exergonic reaction. It releases energy by transferring the third phosphate group from ATP to some other molecule. This transfer is phosphorylation, and these 3 types of cellular work, transport work, mechanical work, and chemical work, depend on ATP energizing molecules by phosphorylating them. So in each of these cases, in the transport work, the mechanical work, and the chemical work, the ATP, via hydrolysis, we can remove a phosphate from the ATP, phosphorylate our membrane protein, our motor protein, or our reactant, depending on which type of work we're doing. And that allows the work to happen. Okay, it allows the work to happen. And then we end up with ADP and our phosphate molecule once it is removed. Okay, so in chemical work, The phosphorylation of reactants provides energy to drive the synthesis of the product. So this phosphorylation drives this formation of these products, the synthesis of the products here. Okay. In mechanical work, like this muscle contraction here, the transfer of the phosphate group to a special motor protein in muscle cells causes the protein to change shape and pull on actin filaments, in turn causing the cells to contract. In this case, you see that contraction happen with the protein moving. Okay. In chemical work, in transport work, we did chemical work. In transport work, like up here, ATP drives the active transport of solutes across a membrane against their concentration gradient. So look at the movement here of this solute across the membrane against the concentration gradient by phosphorylating certain membrane proteins. Okay, so that provides the energy needed to work against the concentration gradient. Here's an example of cellular work that is powered by ATP. Molecular motors move organelles around the cell. The motor proteins require ATP for movement. So this ATP, through hydrolysis, can remove one of the phosphate groups, and through phosphorylation of this motor protein, we can drive the movement of the organelle here forward. Here's another example of cellular work that is powered by ATP. There are pumps in cell membranes that move sodium ions out of cells. So the sodium ion is this little hexagonal shape, so we need to move it out of the cell. How does ATP do that? We phosphorylate the membrane protein and we release energy, and in doing so we can push these sodium ions out of the cell, and we're left with this ADP and a phosphate here. Atp Adp Recycling. ATP is a renewable resource that is regenerated by the addition of a phosphate group to ADP. The energy to phosphorylate ADP comes from catabolic reactions in the cell. The chemical potential energy temporarily stored in ATP drives most cellular work. The Atp Adp cycle occurs constantly in every living cell. It occurs in each of us about 10 to the 21 times per second, or 1 with 3, 6, 9, 12, 15, 18, 21 zeros after it, times per second. These energy conversions are required for life. ATP, ADP, and phosphate are constantly being recycled and reused. Here's a little cycle that shows this regeneration of ATP. So if I have ADP and a phosphate, PI is the abbreviation for the phosphate group. Energy provided from catabolism, which involves the breakdown of nutrient molecules into usable forms. Those are exergonic. They yield energy. So energy from those processes put into the cycle at this point in order to reattach this phosphate group to create ATP. And then this ATP has some energy stored in it that can then be released for cellular work or those endergonic energy consuming processes. And then the cycle repeats itself.